Hey, sleepers. Tonight, we're exploring the quiet dream of forever. The world outside your window is hushed now. Streetlights cast long, soft shadows. The moon hangs in the sky, a silent silver clock that has measured out eons. It has watched mountains rise and crumble. It has seen forests turn to dust and back to forests again. It is a witness to the endless, beautiful, and sometimes fleeting dance of life. And tonight, we are going to gently pull back the curtain on one of life's most profound mysteries. We are going to talk about the idea of not ending, of living on, of immortality. So before you get comfortable, take a moment to like the video and subscribe. It's a small click, but it helps our little community of late-night thinkers grow. And while you're at it, we'd love to know where you're listening from. Tell us your city or country and what time it is there in the comments below. It's always so wonderful to imagine all of us scattered across the globe, sharing this quiet moment together. So close your eyes, take a deep breath, and get ready to drift off as we explore the timeless secrets of immortality. Let's begin not with humans or myths or futuristic machines, but in the deep, dark, silent world of the ocean. Imagine you are floating in a warm, gentle current. The water is a deep, velvety blue. All around you, tiny sparks of bioluminescence drift by like fallen stars. Here, in this peaceful abyss, lives a creature no bigger than your pinky nail. It's called Tortopsis dorni, a rather grand name for such a tiny thing. We know it better as the immortal jellyfish. Now, when we say immortal, we don't mean it can't be harmed. If a fish eats it, well, that's the end of the story for that particular jellyfish. It's not invincible, but it possesses a trick that is, for all intents and purposes, a biological form of time travel. Most creatures are born, they grow up, they reproduce, and then they age and pass away. It's a one-way street, a story with a beginning, a middle, and an end. The immortal jellyfish, however, has found a way to turn the page back. When it gets old, or stressed, or injured, it can do something remarkable. It can revert. It can sink to the ocean floor, and its cells begin to change. Its bell, the floaty part, and its tentacles, the dangly parts, are reabsorbed. It transforms back into a little blob, a cyst. From this cyst grows a polyp, which is the baby stage of a jellyfish. It's like a tiny sea anemone-like stalk. And from this polyp, new, genetically identical jellyfish bud off. An old, mature jellyfish can effectively become a baby again. It's as if a butterfly, upon feeling the end of its days, could transform back into a caterpillar and start its life all over. This isn't just healing. It's a complete do-over. A biological reset button. Scientists call this process transdifferentiation. It's a fancy word for one type of specialized cell, like a muscle cell, turning into a completely different type of specialized cell. It's a superpower that allows this tiny creature to potentially cycle through its life indefinitely, escaping death by old age forever. A silent, pulsing, living symbol of renewal, floating endlessly in the dark. And it's not alone in its defiance of aging. Let's move from the deep ocean to a quiet freshwater pond. Here, attached to a leaf or a stone, you might find another tiny marvel. This one is called the Hydra vulgaris. It looks like a little tube with a crown of waving tentacles. A very simple creature, but its simplicity hides a profound secret. The Hydra doesn't seem to age at all. Scientists have kept hydras in labs for years, watching them, waiting for them to show signs of getting old. They don't. Their mortality rate, their chance of dying on any given day, remains the same whether they are a week old or a decade old. This is because the Hydra is made almost entirely of stem cells. Think of stem cells as your body's master building blocks. They are blank slate cells that can become any type of cell you need. A skin cell, a heart cell, a brain cell. In our bodies, most of our stem cells are tucked away in specific places, and their activity slows down as we get older. But in the Hydra, these stem cells are constantly dividing, constantly replacing old or damaged cells. Its whole body is in a state of perpetual self-renewal. Every few weeks, it has essentially built itself a brand new body. It is forever young, a tiny living fountain of youth in a drop of pond water. One scientist, Daniel Martinez, famously kept a population of them for years, concluding he might die before they showed any sign of aging. A rather humbling thought, isn't it? A creature with no brain, no heart, no eyes, has figured out something that has eluded the greatest minds in human history. Our tour of nature's immortals doesn't stop there. Let's now imagine a shallow stream, the water cool and clear over smooth pebbles. Here you might find a flat, unassuming creature called a planarian flatworm. These little guys are the undisputed champions of regeneration. If you cut a planarian in half, you don't get one dead worm. You get two new, perfectly healthy worms. You can cut them into hundreds of tiny pieces, and as long as a piece is large enough to contain a few of its special stem cells, called neoblasts, it can regrow an entire new body. Head, tail, brain, everything. This incredible ability is powered by an enzyme we're going to talk about a lot more later. It's called telomeres. For now, just think of it as a magical repair kit for its DNA. Because these flatworms keep this repair kit running their whole lives, they can keep their cells young and dividing, allowing for this near-limitless regeneration. It's led to a few jokes in biology labs. Imagine a planarian with two heads after a botched cut. Do they argue about which way to go? Probably not, but it's a fun image. This simple worm shows us that the instructions for rebuilding an entire body can be maintained and used over and over again. Finally, let's leave the water and stand on solid ground, in a windswept, high-altitude mountain range. Here, clinging to the rocky soil, are trees that look ancient. Their trunks are gnarled and twisted by thousands of years of wind and weather. These are the bristlecone pines. Some of them are over 5,000 years old. They were seedlings when the great pyramids of Egypt were being built. They were already ancient when the Roman Empire rose and fell. And the most amazing thing about them is that they show no signs of aging. A 5,000-year-old bristlecone pine is just as healthy and fertile as a 100-year-old one. It still produces seeds. Its wood is still strong. Its needles are still green. They don't die of old age. They die from external factors, a lightning strike, a devastating fungus, a change in climate. They are a testament to endurance, showing us that for some living things, there is no pre-programmed limit, no built-in clock that says, your time is up. They just keep going, slowly, patiently, until the world itself stops them. So we have the jellyfish that can become a baby again, the hydra that is always new, the flatworm that can rebuild from a speck, and the tree that has watched human civilization rise from its infancy. Nature, it seems, is full of creatures that have found a way around aging. This begs a rather important question. If they can do it, why can't we? What is happening inside our own bodies? at a level so small we can't see it. It makes our own clock tick forward, always forward, with no way to turn it back. To understand this, we have to shrink ourselves down past the skin, past the muscle, past the bone. We need to go inside one of our own cells. Imagine a vast, bustling city, full of tiny machines working constantly to keep you alive. And inside the city hall, the nucleus of the cell, are the most important blueprints of all, your chromosomes. These are tightly coiled threads of DNA, the instruction manual for building and running you. Now, look at the very, very end of each of these threads. You'll see a little protective cap. It's like the plastic tip on the end of a shoelace that stops it from fraying. These caps are called telomeres, and they are at the very heart of the story of why we age. Every time one of your cells divides to make a new cell, to heal a cut, or replace old skin, or grow that instruction manual of DNA, has to be copied. But the copying machine inside our cells isn't perfect. Each time it makes a copy, it can't quite get to the very, very end of the thread. So a tiny piece of that protective cap, the telomere, is lost. The shoelace gets a little bit shorter. This happens every single time the cell divides. The telomere gets shorter and shorter and shorter. For a while, this is fine. The cap is just there to protect the important instructions in the middle. But eventually, after about 50 to 70 divisions, the telomere is worn down almost completely. The shoelace is about to fray. The cell knows this is dangerous. If it were to divide again, it might start losing important parts of the DNA blueprint, which could lead to all sorts of problems. So the cell does the sensible thing. It stops dividing. It retires. This limit, the point at which a cell can no longer divide, is called the Hayflick limit, named after the scientist Leonard Hayflick, who discovered it in the 1960s. So, our telomeres act as a kind of molecular clock, counting down the number of times a cell can divide. As more and more of our cells reach this limit and stop dividing, our body's ability to repair and renew itself slows down. This is a fundamental part of the aging process. Now you might be thinking, but what about the hydra and the flatworm? Why don't their shoelaces get shorter? That's because they have that magical repair kit we mentioned earlier, the enzyme called telomerase. Telomerase is like a tiny machine whose only job is to go to the end of the DNA and add back the little bits of the telomere that were lost during copying. It keeps the protective cap long and healthy, so the cell can keep dividing indefinitely. The discovery of this enzyme was so important that the scientists who figured it out, Elizabeth Blackburn, Carol Greider, and Jack Szostak, won the Nobel Prize for it. So why don't all our cells use telomerase all the time? Well, some of them do. Our reproductive cells, for instance, need to be able to divide many times without aging to pass on our genetic information to the next generation. But in most of our adult body cells, the gene for making telomerase is switched off. The reason is a kind of trade-off, an incredibly important one. Uncontrolled, endless cell division is the hallmark of cancer. One of the first things a cancer cell often does is figure out how to switch the telomerase gene back on, giving it a form of cellular immortality. So, our bodies suppress telomerase in most cells as a powerful defense mechanism against cancer. We trade a limitless lifespan for a lower risk of tumors. It's a profound biological bargain that is written into our very cells. But telomere shortening isn't the only story. There's another character in this cellular drama. Sometimes a cell becomes damaged. Maybe it's been hit by too much radiation from the sun, or it's been stressed by toxins. The cell's internal quality control systems sense that something is wrong. If the damage is too great to repair, the cell has two options. It can trigger a self-destruct sequence, a process called apoptosis, or programmed cell death. Or it can enter a state of suspended animation. It doesn't die, but it permanently stops dividing. This is called cellular senescence. Think of these senescent cells as zombie cells. They are still hanging around in our tissues, but they are not doing their jobs properly. And worse, they start secreting a cocktail of inflammatory chemicals that can damage the healthy cells around them. It's like one grumpy, retired worker sitting in the middle of a busy office, complaining loudly and distracting everyone else. When we are young, our immune system is very good at finding and clearing out these zombie cells. But as we get older, our immune system becomes less efficient. These senescent cells start to accumulate in our tissues, throughout our body, in our skin, our kidneys, our hearts, our brains. And the chronic, low-grade inflammation they cause is now thought to be a major driver of many age-related diseases. And finally, there's the simple wear and tear of daily life. Our DNA, that precious instruction manual, is under constant assault. From things outside us, like UV radiation from the sun, and from things inside us, like highly reactive molecules called free radicals, which are byproducts of the very process of turning food into energy. Every day, the DNA in each of your cells suffers thousands of damaging events. Most of the time, the cell's sophisticated repair crews fix the damage perfectly, but they aren't flawless. Over time, some damage slips through. A letter in the genetic code might get changed. A tiny smudge appears on the blueprint. Over decades, these little errors accumulate. The instruction manual gets a little harder to read. The cell might start to malfunction. This accumulation of DNA damage is another core reason why our bodies gradually decline with age. So it's a combination of things. Our cellular clocks ticking down, the rise of zombie cells, and a lifetime of accumulated damage to our most fundamental code. This is the biological reality of aging. But the dream of escaping this reality is as old as humanity itself. Long before we knew about cells or telomeres, we were telling stories and searching for ways to live forever. Let's travel back in time to the land of ancient Egypt under a hot, brilliant sun. The Egyptians had a profound and complex vision of the afterlife. They believed that death was not an end, but a transition. To make this journey, however, the body had to be preserved. They believed a person had several souls, or aspects of their being, and one of them, the Ka, needed to be able to recognize the body to return to it. This belief gave rise to the intricate and solemn practice of mummification. It was a painstaking process, taking 70 days. They would remove the internal organs, dry out the body with a special salt called natron, and then wrap it in hundreds of yards of linen bandages. It was a form of applied immortality, a technology designed to ensure the body's integrity for an eternal existence in the next world. It was a testament to how deeply the desire for continuity is woven into our consciousness. Now, let's journey east to ancient China. Around 200 BCE, we meet a powerful and formidable figure, Qin Shi Huang, the first emperor of a unified China. He was a man of immense ambition. He built the Great Wall and the incredible terracotta army, thousands of life-sized clay soldiers to guard him in the afterlife. But he wasn't content with just a glorious afterlife. He wanted to conquer death itself. He sent expeditions far and wide in search of a mythical elixir of life. His court alchemists and physicians worked tirelessly to create potions that would grant him immortality. Tragically, one of the key ingredients in many of these supposed elixirs was mercury. We now know mercury is a potent neurotoxin. It's poison. The emperor, in his desperate quest for eternal life, consumed these mercury pills. It's widely believed that this quest for immortality is what ultimately killed him at the relatively young age of 49. It is one of history's most poignant ironies. The search for forever can sometimes hasten the end. This idea of a magical substance that could grant eternal life persisted for centuries. In medieval Europe, it took the form of the philosopher's stone. This was the ultimate goal of the alchemists, a legendary substance that could not only turn base metals like lead into pure gold, but could also be used to create an elixir of life, granting immortality to whoever drank it. Alchemists like the famous Nicholas Flamel, who lived in the 14th century, were rumored to have discovered the stone and achieved immortality. Of course, these are just legends, but they show how the dream of gold and the dream of eternal life were intertwined. Both represented a kind of perfection, a transcendence of the ordinary, decaying world. The alchemists were not just greedy men trying to get rich. They were philosophers and mystics, trying to understand the fundamental nature of reality and unlock its deepest secrets, including the secret of life itself. And what about the Fountain of Youth? That story is most famously linked to the Spanish explorer Juan Ponce de Leon. The legend says that in 1513, he led an expedition to what is now Florida, searching for a mythical spring whose waters would restore youth to the old. It's a romantic and enduring image. Interestingly, historical records show that Ponce de Leon was primarily on a mission for gold and land, under a charter from the Spanish crown. The Fountain of Youth story was actually attached to his name by a writer decades after his death. But the myth stuck because it tapped into such a powerful, universal desire. The idea that somewhere in a hidden, unspoiled corner of the world, there exists a simple, natural cure for aging. A quick dip in a magical pool, and all your troubles are washed away. It's a beautiful, simple fantasy, and one that we are still chasing in a way with every new vitamin, skin cream, and health fad that promises to turn back the clock. While these historical quests for a magical cure were ultimately fruitless, something else was happening in the background. Slowly, painstakingly, humanity was actually succeeding in extending life. Not through elixirs or fountains, but through knowledge, observation, and cooperation. Let's look at the incredible journey of human life expectancy. In the year 1900, the average person on Earth could expect to live to be about 31 years old. Today, the global average is over 72 years. That's more than double. This is one of the most astonishing achievements in the history of our species. And the biggest reasons for this leap are, perhaps surprisingly, not the high-tech medical marvels we might imagine. The single biggest contributor, especially in the early days, was the dramatic reduction in infant and child mortality. In 1900, it was tragically common for children to die from diseases that are now easily preventable. Things like diarrhea, measles, and respiratory infections. The heroes of this story are not just famous scientists, but the engineers and public health officials who brought us sanitation systems and access to clean drinking water. Simply separating sewage from the water people drank prevented countless deaths from diseases like cholera and typhoid. It's not glamorous, but it saved more lives than almost any other invention until the mid-20th century. Proper hygiene, understanding how germs spread, and basic nutrition made it possible for millions more children to survive to adulthood. Then came the age of medical miracles. In 1928, a Scottish scientist named Alexander Fleming was tidying up his lab. He noticed that on a petri dish he'd forgotten about, a patch of mold was growing. And all around that mold, the bacteria he had been trying to grow were dead. He had stumbled upon penicillin, the world's first antibiotic. It took another decade to purify it and turn it into a medicine, but its impact was revolutionary. For the first time, we had a powerful weapon against bacterial infections that had plagued humanity for millennia. Infections from a simple cut, or during childbirth, or from diseases like pneumonia, which were often death sentences, could now be cured. Millions of lives were saved. Around the same time, we were perfecting another powerful tool: vaccines. The concept had been around since the 18th century, with Edward Jenner's pioneering work using cowpox to protect against smallpox. But in the 20th century, we developed vaccines for a whole host of devastating diseases. Polio, which could paralyze children for life. Measles, mumps, rubella, tetanus. The principle is simple and elegant. A vaccine introduces a safe, weakened, or inactivated part of the germ to your immune system. Your immune system learns to recognize it and builds an army of antibodies to fight it. Then, if you are ever exposed to the real dangerous germ, your body is already prepared. It knows the enemy and can defeat it quickly. Thanks to vaccination campaigns, we have completely eradicated smallpox from the planet, and we are on the verge of doing the same with polio. These interventions have added decades to the average human lifespan. We have extended life not by stopping aging, but by preventing premature death. We have given more and more people the chance to live out their full natural lifespan. And now, the scientific quest has shifted. The question is no longer just, how do we stop people from dying young? But can we slow down, or even reverse, the aging process itself? To answer that, we have to go back to our DNA, our genetic blueprint. We are all built from a similar set of instructions, but there are tiny variations from person to person. And some of these variations seem to be linked to exceptional longevity. Scientists studying groups of centenarians people who live to be 100 or older have found that they often share certain genetic traits. One gene that comes up again and again is called FOXO3. You can think of FOXO3 as a kind of master supervisor for cellular maintenance. Certain versions of this gene are better at protecting cells from stress, repairing damaged DNA, and keeping the immune system in good shape. People with these beneficial variants of FOXO3 are significantly more likely to live a very long and healthy life. This has been found in populations all across the world, from Japanese people in Okinawa to Germans to Ashkenazi Jews, suggesting it's a fundamental part of the human longevity toolkit. Other genes are involved too. For example, many centenarians have protective versions of genes that control cholesterol metabolism, like a POE and CETP. These variants help keep their arteries clear and their cardiovascular systems healthy well into old age. It's like being born with a car engine that is naturally better at staying clean and running efficiently. This doesn't mean there is a single immortality gene. It's more like having a winning combination of many small genetic advantages that, together, give your body a greater resilience against the ravages of time. We can also learn a great deal about aging by studying the opposite, diseases of accelerated aging. These are rare genetic conditions called progeria syndromes. The most well-known is Hutchinson-Gilford progeria. Children with this condition have a tiny mutation in a gene called LMNA. This gene is supposed to provide the structural scaffolding for the cell's nucleus. The mutation makes this scaffolding unstable. As a result, these children age at a breathtakingly rapid pace. They lose their hair, their skin wrinkles, and they develop diseases like arthritis and heart disease, all within the first decade of their life. They typically live only into their early teens. It is a heartbreaking condition. But by studying what goes wrong in these children, scientists can gain crucial insights into the normal aging process. Understanding why the unstable nucleus in progeria causes such rapid aging helps us understand how a stable nucleus protects us during normal aging. It's like learning how a building stands by studying why another one collapses. And once again, nature has some quirky examples to teach us. Let's meet one of the most bizarre and wonderful mammals on the planet, the naked mole rat. These are wrinkly, buck-toothed, sausage-shaped rodents that live in underground colonies in Africa. A mouse or a rat of a similar size might live for 2 or 3 years. The naked mole rat can live for over 30 years. That is a 10-fold increase in lifespan. They are also incredibly resistant to cancer and seem to feel very little pain. They are, in many ways, the superheroes of the rodent world. So what's their secret? Scientists are still figuring it out, but they've found some clues. Their proteins, the tiny machines that do all the work in their cells, are uniquely stable and resistant to misfolding, a common problem in aging cells. They also have a special kind of sugar molecule in their tissues that seems to protect them. The naked mole rat is a living laboratory, showing us that even among mammals, there are wildly different ways to age, and that with the right biological tricks, a long and healthy life is possible. Inspired by these clues from genetics and nature, scientists today are in a race to develop interventions that could slow down aging in humans. This is no longer the realm of science fiction. It's happening in labs all over the world. One of the most promising molecules being studied is called rapamycin. It was first discovered in a soil sample from Easter Island, which is also known as Rapa Nui, hence the name. Rapamycin works by inhibiting a pathway in our cells called mTOR. You can think of mTOR as the cell's growth and metabolism dial. When mTOR is turned up high, the cell is in growth mode. It's building new proteins, dividing, and using lots of energy. This is great when you're young and growing, but having the dial cranked up all the time in adulthood seems to accelerate aging. Rapamycin gently turns down the mTOR dial. It tells the cell to slow down, to stop focusing on growth, and start focusing on maintenance and repair. In countless studies, giving rapamycin to mice has extended their lifespan by up to 25%. That would be like adding 20 years to a human life. It's one of the most robust life-extending interventions ever discovered in a mammal. Another exciting candidate is a drug that millions of people already take every day. It's called metformin. It's a safe, cheap, and common drug used to treat type 2 diabetes. But researchers noticed something odd. People taking metformin for their diabetes seem to be living longer, on average, than healthy people who didn't have diabetes at all. They also had lower rates of cancer and heart disease. Metformin seems to work by gently nudging our metabolism to a more youthful state, similar to the effects of exercise or calorie restriction. The evidence is so compelling that a major clinical trial is now underway, called the TAME trial, which stands for Targeting Aging with Metformin. It will be the first trial of its kind to test a drug not for a specific disease, but for its ability to slow down the overall process of aging in humans. If it's successful, it could fundamentally change how we practice medicine. Many of these anti-aging strategies revolve around energy. As we get older, our cells get worse at producing energy. The little power plants in our cells, the mitochondria, start to break down. A key molecule involved in this energy production is called NAD+. Levels of NAD decline dramatically as we age. It's like the battery in our cells is slowly running down. So, a lot of research is focused on boosting NAD+ levels. Supplements like NMN and NR are precursors to NAD, meaning our bodies can use them as raw materials to make more. In animal studies, boosting NAD has shown remarkable results, restoring mitochondrial function, improving muscle strength, and even making old mice look and act younger. Human trials are still in their early days, but it's a very exciting area of research aimed at quite literally recharging our cells. And what about those zombie cells we talked about earlier? The senescent cells that accumulate and spread inflammation. What if we could just get rid of them? This has led to the development of a whole new class of drugs called senolytics. These are drugs that can selectively seek out and destroy senescent cells while leaving healthy cells unharmed. It's like a smart bomb for a specific aspect of aging. In animal studies, giving a senolytic cocktail, like the combination of a cancer drug called dasatinib and a plant-based supplement called quercetin, has had incredible effects. It has improved heart function, reduced osteoporosis, and extended the health span of old mice, making their remaining time more active and disease-free. Several senolytic drugs are now being tested in human clinical trials for age-related conditions like arthritis and lung disease. The idea of a periodic zombie cell cleanse to rejuvenate our tissues is a very powerful one. Beyond just slowing down aging, there is an even more ambitious field, regenerative medicine. The goal here is not just to maintain what we have, but to rebuild what has been lost. The cornerstone of this field is the stem cell. In 2006, a Japanese scientist named Shinya Yamanaka made a discovery that would change the world and earn him a Nobel Prize. He discovered a way to take an ordinary adult cell, like a skin cell, and reprogram it. He found a cocktail of just 4 genetic factors that, when added to the cell, could turn back its developmental clock. The adult skin cell would become a stem cell again, a cell that has the potential to become any other type of cell in the body. These are called induced pluripotent stem cells, or iPSCs. This discovery was revolutionary. It means we could theoretically take a small sample of your own skin, create stem cells from it, and then coax those stem cells to grow into new heart muscle to repair a damaged heart, or new neurons to treat Parkinson's disease, or new retinal cells to cure blindness. All using your own cells, so there would be no risk of your immune system rejecting them. The work is still highly experimental, but it holds the promise of true regeneration. But what if you need a whole new organ? Building a complex, 3-dimensional organ, like a kidney or a liver, is an incredible engineering challenge. This is where 3D bioprinting comes in. It works a bit like a regular 3D printer, but instead of using plastic, it uses a bioink made of living cells and a scaffold material. Layer by layer, the printer can build up a complex tissue structure, guided by a computer model. Scientists have already successfully printed and implanted simpler structures, like skin, cartilage, and even functional bladders, into human patients. The dream is to one day be able to print custom-made, fully functional organs on demand, ending the long and often tragic waiting lists for organ transplants. Another powerful tool in the regenerative toolbox is gene editing. A technology called CRISPR, in particular, has made it possible to edit our DNA with incredible precision. You can think of it as a find and replace function for our genetic code. Scientists can guide the CRISPR system to a specific gene, and it can then cut out a faulty piece of DNA and replace it with a correct one. This could be used to correct the genetic mutations that cause diseases like cystic fibrosis or Huntington's. But in the context of aging, it could also be used to fix accumulated DNA damage, or even to enhance our natural protective genes, perhaps giving us a more beneficial version of that FOXO3 gene we talked about. The ethical implications of editing our own genetic code are profound and complex, but the potential to cure disease and enhance resilience at the most fundamental level is undeniable. And then there's a stranger, more controversial area of research that came from a series of rather macabre-sounding experiments. The technique is called parabiosis. It involves surgically joining two animals, usually mice, so that they share a common circulatory system. Their blood flows between them. When scientists joined an old mouse to a young mouse, they observed something astonishing. The old mouse started to show signs of rejuvenation. Its muscles became stronger, its brain function improved, and its tissues started to heal more like those of a young mouse. Conversely, the young mouse started to age faster. This suggests that there are factors in the blood of young animals that have powerful regenerative properties, and factors in the blood of old animals that promote aging. Scientists are now trying to identify these specific youth factors and age factors in the blood. The goal is not to connect old people to young people, of course, but to create a drug or a therapy that can mimic the rejuvenating effects of young blood without all the obvious practical and ethical problems. But what if all these attempts to repair the biological body fail? What if, despite our best efforts, the end still comes? For some, this is where the idea of cryonics comes in. Cryonics is the practice of preserving a legally deceased person at extremely low temperatures, in liquid nitrogen, with the hope that future medical technology will be able to revive them, repair the damage that caused their death, and reverse the aging process. It's a bet on the future. A very big bet. It's important to be clear. This is not freezing. If you just froze a body, the water in the cells would form sharp ice crystals, which would shred the cell structures to pieces. Instead, cryonics uses a process called vitrification. The blood is replaced with a cryoprotectant solution, a kind of medical-grade antifreeze. The body is then cooled very rapidly to a temperature of around minus 196 degrees Celsius. At this temperature, the molecules essentially stop moving, and the body can be preserved indefinitely without decay. The cryoprotectant solution doesn't form ice crystals. Instead, it turns into a smooth, glass-like solid, which keeps the delicate cellular structures intact. As of today, there are over 500 people who have been cryopreserved around the world, with thousands more signed up. But there is a huge, unavoidable catch. We have absolutely no technology to revive a cryopreserved person. None. The people who choose cryopreservation are placing their faith in the idea that future civilizations will not only be incredibly advanced medically, but will also be willing and able to revive them. The challenges are immense. They would need to warm the body without damaging it, reverse the toxicity of the cryoprotectant, repair the cellular damage from the original freezing and thawing process, cure the disease that originally caused the person's death, and reverse the aging that had occurred throughout their life. It is a monumental task. The cost is also significant, ranging from tens of thousands to hundreds of thousands of dollars, raising profound ethical questions about who could afford this potential shot at a second life, and whether it's a wise use of resources. It remains a deeply speculative, but undeniably fascinating intersection of hope, technology, and faith in the future. If preserving the body seems too difficult, what about preserving the mind? This leads us to the concept of digital immortality. The most famous idea here is mind uploading. The theory is that if consciousness is purely a product of the brain's structure and patterns of activity, then if we could create a perfect atom-for-atom scan of a brain and simulate it on a powerful enough computer, we could effectively transfer a person's consciousness from their biological brain to a digital substrate. Your mind, your memories, your personality would live on as a program running on a computer, free from the frailties of the body. You could live in a simulated virtual reality, or perhaps even pilot a robotic body. It's an idea straight out of science fiction, but some scientists take it very seriously. Projects like the Blue Brain Project have been working for years to create a detailed digital simulation of a piece of a rat's brain. By 2015, they had managed to map a single cortical column containing about 31,000 neurons. It was a huge achievement, but the full human brain has around 86 billion neurons. The scale of the challenge is almost incomprehensible. The computational power required to simulate a full human brain in real time is estimated to be many orders of magnitude beyond what our most powerful supercomputers can do today. And that's before we even get to the biggest question of all. Is consciousness something that can actually be copied? Would the simulation on the computer truly be you? Or would it just be a perfect replica, an echo, while your own consciousness fades away with your biological brain? It's a philosophical question that we have no way of answering right now. A more immediate, and perhaps more unsettling, form of digital immortality is already here. There are now companies that can take a deceased person's digital footprint, their emails, their social media posts, their text messages, and use it to train an AI chatbot. This chatbot can then mimic the person's personality and writing style, allowing their loved ones to have conversations with a digital avatar of the person they lost. This raises a whole host of ethical concerns. Does the deceased person have a right to be forgotten? Did they consent to having their personality recreated in this way? Can interacting with a digital ghost be psychologically healthy for those who are grieving? It's a strange new territory for humanity, where our digital echoes might outlive our physical selves. We are already creating the first ghosts in the machine. Whether we are trying to repair the body, freeze it, or upload it, all these futuristic ideas are a long way off. But what can we do right now with the knowledge we have? One of the most well-researched and powerful ways to extend not just lifespan, but health span is surprisingly simple. It's caloric restriction. For almost a century, scientists have known that reducing the amount of calories an animal consumes without causing malnutrition can dramatically extend its life. This has been shown in everything from yeast and worms to flies, mice, and even primates. In many species, a 30 to 40% reduction in calories can lead to a 30 to 40% increase in lifespan. The reasons are complex, but it seems that when the body is in a state of scarcity, it switches from growth mode to survival and repair mode. It becomes more efficient at clearing out damaged cells and protecting itself from stress. One of the key processes that caloric restriction activates is called autophagy. You can think of autophagy as the cell's internal recycling and cleanup crew. The cell identifies old, damaged, or misfolded proteins and other cellular junk, breaks them down into their basic building blocks, and then recycles those building blocks to make new, healthy parts. It's a form of cellular spring cleaning. This process naturally declines as we age, leading to a buildup of cellular garbage. Caloric restriction and its more popular cousin, intermittent fasting, both give a powerful boost to autophagy, helping to keep our cells clean and functional for longer. Of course, long-term human studies on caloric restriction and lifespan are difficult to conduct. They would take over a century. But shorter-term studies, like the calorie trial, have shown that even a modest 15% reduction in calories in healthy humans leads to significant improvements in biomarkers of aging, like lower blood pressure, better cholesterol levels, and reduced inflammation. It seems that eating a little less might be one of the most powerful things we can do to live a little longer and a little healthier. So, with all this research, from genetics to drugs to diet, what is the realistic limit of a human life? Is there a hard ceiling we can't break through? The official, verified record for the longest-lived human belongs to a French woman named Jeanne Calment. She was born in 1875 and passed away in 1997 at the incredible age of 122 years and 164 days. She lived through the invention of the automobile, the airplane, 2 world wars, and the birth of the internet. She even met Vincent van Gogh when she was a young girl. For decades, her record has remained unbroken. This has led some scientists to suggest that the maximum human lifespan might be biologically fixed, somewhere around 115 to 125 years. Statistical analysis of global demographic data seems to support this, showing that while the average life expectancy has soared, the maximum age of death has not really increased much since the 1990s. However, other researchers disagree. They point to an interesting quirk in mortality data. For most of our lives, our risk of dying doubles approximately every 8 years. But when you look at supercentenarians, people who live past 110, this acceleration seems to stop. Their mortality rate plateaus. A person who is 112 has roughly a 50/50th chance of making it to their next birthday, and a person who is 117 has roughly the same 50/50th chance. It doesn't seem to get any worse. This suggests that if we can overcome the frailties of our 80s and 90s, we might enter a new phase of late life survival where there is no predetermined stopping point. And as medicine continues to advance, and the number of people living to 100 explodes, it's projected there will be over 1 million centenarians worldwide by 2050. It seems almost inevitable that Jeanne Calment's record will one day be broken. The question is, by how much? This brings us to the final, and perhaps most important, set of questions. What if we succeed? What if we develop technologies that allow people to live for 150, 200, or even 500 years? What would that world look like? The ethical and social implications are staggering. The first and most obvious concern is overpopulation. If people stop dying at the rates they do now, but we continue to have children, our planet's resources would be strained to the breaking point. This would likely require profound societal changes in our approach to family and reproduction. Then there is the issue of inequality. Life-extending therapies, at least initially, would likely be incredibly expensive. This could create a world starkly divided into two classes: a wealthy elite who can afford to live for centuries, and the rest of humanity who live and die by the old rules. A world of immortal elites and mortal commoners. This could exacerbate social tensions and create forms of inequality we can barely imagine. Our entire social structure is built around a lifespan of about 80 years. How would a world of 200-year-olds function? Pension systems designed to support people for 15 or 20 years of retirement would completely collapse. People might have to work for over a century. The concept of education would have to change. You might go back to university for a completely new career at the age of 100. And what about our relationships? Could a marriage endure for 3 centuries? How would you relate to your great-grandchildren? And then there are the psychological challenges. Our brains are not designed to store hundreds of years of memories. Would we forget our early lives? Would we become trapped by the grief of outliving everyone we have ever loved, generation after generation? Would we become endlessly bored, apathetic, and resistant to change? The very meaning of life, which is so often shaped and given urgency by its finiteness, would be thrown into question. What is the purpose of a single day when you have 100,000 more ahead of you? These are not easy questions, and they are questions we need to start thinking about now, before the science fiction becomes scientific fact. So, where does that leave us? What is the current scientific consensus on immortality? Today, most mainstream researchers in the field of aging have shifted their focus. The goal is not so much to extend lifespan, the total number of years we live, but to extend health span, the number of years we live in good health, free from chronic disease and disability. The idea is to compress morbidity, to squeeze the period of late life illness and frailty into a much shorter time. The dream is to be healthy and active at 95, and then to have a rapid decline, rather than a slow, decade-long deterioration. A major turning point in this effort came recently when the World Health Organization, in its International Classification of Diseases, the ICD-11, added a new code, aging-related. This officially recognizes aging itself as a condition that can be targeted for prevention and treatment. It's a subtle change in language, but it unlocks the ability for pharmaceutical companies to develop and test drugs specifically for aging, just like they do for cancer or heart disease. It legitimizes the entire field. The modern scientific framework for this work is a landmark 2013 paper that outlined the hallmarks of aging. It identified 9 distinct categories of cellular and molecular damage that drive the aging process things. We've talked about like telomere shortening, cellular senescence, and mitochondrial dysfunction. This paper provided a roadmap for researchers. It said, these are the targets. If we can develop interventions that address each of these hallmarks, we can slow down and perhaps even reverse the aging process. And so, the quiet, painstaking work continues. Most serious researchers in the field would tell you that true biological immortality for humans is unlikely to be achieved anytime soon, if ever. The complexity of our biology is simply too vast. But many are cautiously optimistic that significant life extension is achievable within this century. The idea of an average, healthy lifespan of 120 or even 150 years is no longer considered fantasy. It is a tangible goal that is being pursued in labs all around the world. We may not live to see forever, But we may live to see a world where our children and grandchildren have the gift of many more healthy decades, many more years to learn, to love, to create, and to experience the profound and beautiful mystery of being alive. And as you drift off to sleep tonight, perhaps you can dream of that future, a future where the clock still ticks, but perhaps just a little more slowly.